Hi, Dr. Sinerik. If you would please let me, I will introduce you and then you can start your talk. I should give a quick review of you so that everyone would get to know you. Yes. Okay, already welcome everyone on behalf of everyone at Confrontiers team. I thank you all our brilliant participants for their time and support. My name is Nafisa Norouzi. I will give you my presentation later on today, but for now I'm your moderator for this event. And I would like to remind everyone, in order to stay on the schedule. In order to stay on the schedule, please keep in mind that your talk will be within 30 minutes tops and not less or more. And please, everyone, please turn on your videos during the discussion after every talk. Okay, so we are going to start with Dr. Sinerik Ayrapetyan. Professor Sinerik has received his PhD in cell biophysics in the Institute of Physiology of Ukraine Academy of Sciences, Kiev 7. Currently, he is the coordinator of UNITwin Postgraduate Educational Center, Yerevan, Armenia. His research has included the study of metabolic regulation of cell function in norm and pathology. He is serving as a chief editor for the Journal of Bioequivalence and Bioavailability, and he is an editorial member of several reputed journals like Electromagnetic Biology and Medicine, ISRN Biophysics, European Journal of Biophysics, Advances in Life Sciences, Applied Pharmacy, BBA General Subjects, International Dental and Medical Research, International Journal of Basic and Applied Sciences, Insights of Medical Sciences, Chronicles of Pharmaceutical Sciences Journal, Clinical Investigations and Medical Sciences, the Global Drugs and Therapeutics, and etc. Professor Sinerik is a member of a number of international societies, such as International Society of Invertebrate Neurobiology, International Society of Neurochemistry, European Society for Neurochemistry, International Brain Research Organization, International Union of Pure and Applied Biophysics, Bioelectromagnetic Society, WHO International Advisory Committee on Electromagnetobiology, and the President of All Armenian Research Council. He has authored 7 international books, 115 research articles. The title of his talk today is The Activation of Quantum Sensitive CRM-Pair-Dependent Sodium-Calcium Exchange in reverse mode as a primary mechanism for generation of pain. Please, Dr. Sinerik, please start your
well, thank you very much for your introducing me. It's too longer. Please, next time, make shorter. Okay, let's start. As you know, pain can provocate by different factors, starting mechanical damaging and psychological information and different weak signal environmental medium, which could generate pain or modulate the pain. All these data indicate that it must be distinct mechanism in cell membrane, quantum sensitive mechanism, which are able to receive quantum signals and response it to keep control steady state cell and organism. Still, since in 17th century, Descartes suggests that pain signal from periphery can transmit through central system nervous system and forming pain signal in brain. Unfortunately, now we have a rather rich information about adverse signal effect on cell and organism, and limited knowledge about beneficial signal effect on cell and organism. As you know, in 1963, a group of scientists headed by Alan Hodgkin, they have a Nobel Prize for discovery the ionic mechanisms involved in excitation and inhibition of nerve cell membrane. Unfortunately, that this discovery, it was a revolutionary discovery in to understand the cell membrane in generation electrical activity, but because this theory was developed on intracellular perfused squeeze axon with losing intracellular metabolism. They have a critical defect. Sorry? Please close your
6,000 attendees
microphone.
Invitation. 100.
Please, organizer, close your microphone.
అంటే లిమిట్ ఇట్లా తీసుకోవడం ఉంటుందా అన్నయ్య పెయిడ్ వర్షన్ అయ్యో
Hey, I'm sorry, but please mute your microphones. Thank you.
Okay. And this theory has a critical defect because they're unable to explain metabolic regulation, metabolic controlling semi-permeable properties of the membrane. They're explaining based on classic thermodynamic principles that electrochemical gradient driving ionic current through the membrane is main signal transition pathway. But from this theory, it's very difficult to explain quantum mechanical sensitivity, that membrane excitability, you know, that from biophysical point of view, the pain can be considered as abnormal excitation nerve ending, which transmitting involving in central nervous system, forming as a pain signal. So the second defect of this theory that they didn't consider water flux's effect on ionic current. On this figure, you can see classical experiment, intracellular perfused squeeze axon, left side when in internal and external solution was you have problem? Have you problem to follow my thought?
సరే సరే అండి ఇప్పుడైతే మొత్తం అయితే తీసేయకండి అన్న 90 ఉండండి ఓకే
Can I continue? Hello.
I am sorry, Dr. Sinerik. I don't know whose mic is on. I can't tell them to turn it off. I'm sorry. Please continue. I will ask the host to mute all participants.
Okay, Nafizi. Yeah.
Yes, please, Mr. Karishna, mute everyone except Mr. Sinerik. Yes.
Sorry
Yes,
for that.
thank you so much. Professor Sinerik, please continue. I'm sorry for the interruption.
Thank you. So on this figure, you can see this voltage clamp experiment internally perfused taxon. In left side, when internal and external side is an isotonic solution, you can see inward current and potassium current and tail current. And it's enough to apply water flux
Professor Sinerik, you are muted. Please unmute your audio.
Okay.
Yes, that's better.
Okay.
Now we can hear you,
I'm
Dr.
sorry.
Sinerik. Yeah.
Yes, we can hear you now.
sorry.
Please continue.
Now, can I continue?
Yes, please continue. We can hear you.
Okay. This is the taxon. When water efflux flying, right side, you can see that inhibiting inward sodium current, activated potassium current, and tail current, it's also potassium current, but inward direction and fully eliminated. So these water fluxes did not consider Hodgkin and his group when they study transmembrane current, ionic current in depolarization or hyperpolarization. And after discovering electrogenic sodium pump, which was shown that normal functioning nerve cells pump working in electrogenic regime, and it's been that pump could regulate its single metabolic mechanism which could participate in generation membrane potential, and soon in different laboratory independently was discovered in various subjects that really pump is functioning in normal living state, which was rejected by author of membrane theory. And soon Roger Thomas published physiological review in which now he demonstrated that already the existence electrogenic sodium pump can be considering as proving fact. From this suggests that sodium potassium pump, because working in stoichiometry, pushing out 3 sodium and accumulating 2 potassium, by this way generating osmotic gradients on the membrane, and from other side being high ATP utilizing machine by activation intracellular oxidative phosphorylation stimulate endogen water molecule formation. And it's well known from biochemistry book that one molecule glucose oxidation producing about 42 molar water. And this endogen water by hydration intracellular molecules controlling their activity, and other sides by generation osmotic gradients on the membrane producing water efflux from the cell. So from the right side, you can see that inward water influx activates sodium influx, while pump metabolic driving water efflux from the cell, inhibiting this stability. So dysfunctioning, sustainable dysfunctioning, this impairment of water efflux from the cell brings of a normal excitation neuronal membrane, which can be considered as a primary mechanism of pain signal generation. Okay, and next slide. Okay, it's very easy to demonstrate that pain threshold how it correlate with hydration, brain cortex hydration. In left side, you can see hydration, its first column, its control, and right side, pain threshold, which is detected by a hot plate. And you can see that by decreasing hydration, there is a correlate increasing pain threshold. So decreasing sensitivity organism rats to pain. And second column is 30 minutes microwave, not microwave, mechanical vibration for Hertz. We have shown that it activates sodium potassium pump by pushing calcium from the cell. A recipe was published in Magnetobiology. And this dehydration accompanied by increasing threshold, pain threshold. And also very easy method for injection mannitol, which is osmotic active substances, and by dehydration, again, increasing pain threshold. Okay, but what happens? Let's now, can you follow me?
Yes, Dr. Sinerik, yes.
Okay, let's now consider the cellular mechanism of generation pain. And on this slide, you can see same neuron in pump active in left side and the right side to pump inactive side. And you can see that cell swelling when pump inactive side because of water uptake. What happens in that case? We have shown that protein molecules in cell membrane are functionally active in the reservoir state depending on the membrane surface. So, by cell swelling, it's increasing number of functional active ionic channels, receptor, and pump units. On this slide, you can see that pump activation, even that case when membrane depolarizing in Japanese and land snails has abundance intracellular potassium, high concentration potassium, and small potassium concentration changing extracellular depolarizing. It's well known that potassium activate the pump. As you can see that when pump activated by potassium, increasing up to 0 potassium potassium-free solution, we have depolarization of the membrane, but inhibition spontaneous activity. This is the bursting activity, so-called epileptic discharge of normal endogen activity. It's not synaptical. It's more pronounced if we will replace potassium ion by Orubiji, then this gradient is rather higher, and again you can see that pump activation, in spite that depolarizing membrane, we have inhibition. So by this data, it was clearly demonstrated that pump can regulate excitability cell, not only by hyperpolarization of the membrane as predicted from Hodgkin-Huxley model, but also by generation metabolic water efflux from the cell having inactivation sodium inward current. And what happens with muscles? If it's well known that pump inactivation accompanied by activation calcium influx, and because about 50% of myocyte volume is myosin, it's a high sensitivity for calcium because reciprocal sensitivity to calcium, muscle shrinking and neuronal swelling because pump inactivation, then micro deformation junction between nerve ending and muscle is extracellular second mechanism generating pain signal. So now we understood that really pump, sodium potassium pump could regulate membrane excitability, but it's well known that pain signal is a quantum mechanical sensitive. Even non-thermal intensity microwave can be used for modulation pain. But pump, it's not quantum sensitive. It means that it must be in cell membrane special quantum sensitive mechanism which controlling pump activity. To evaluate the nature of this mechanism, we have studied dose-dependent O-binding binding with extracellular concentration of O-binding. O-binding is a specific inhibitor, sodium-potassium pump, and we find out it our early work in '84 in collaboration with Peter Becker, who is author of perfused quitaxone. We find out that there is a three family of O-binding receptors. Both two of them have dose-dependent saturating, and one of them linear dose-dependent character has, and its highest sensitive O-binding and low affinity O-binding receptors increasing by cell swelling. This is a curve in the middle isotonic solution and hypotonic and hypertonic solution, that by changing surface, all these three family receptors activating. And more detailed investigation of this have shown that two of them, it's not canonical O-binding receptor. It can be activated by extremely weak concentration, homeopathic concentration substances or weak signal, which are unable to activate ionic channels in the membrane and has no effect on pump activity. This linear part is a pump, and this can be seen in next slide. If we study sodium efflux depending on O-binding concentration, you can see this is dashed line in the normal state, and the higher concentration of bioin as predictable inhibit sodium potassium pump, but we find out extremely low concentration of bioin stimulate sodium efflux from the cell, and from the right side is pump activity, which detecting biorubiji uptake, pump activity no changing. This discovery, it's possible only by study whole neurons, and it is absent in squid taxon when push out cytoplasma from the cell. So if we will study same thing with acetylcholine concentration, acetylcholine, you know that is transmitters, excitative transmitters, and again, this dose-dependent effect acetylcholine on sodium efflux. You can see same picture as in case of bioin. Again, low concentration acetylcholine has to stimulate sodium efflux from the cell, while just high concentration, which activate ionic channels, then we have sodium efflux into the cell. And this is, you can see this voltage clamp experiment in 10-6 molar acetylcholine induced current. If we will apply just 10-8 molar peroxide, you can see that depressing this. But peroxide itself has no any effect on membrane conductivity. And when wash out, again returning back. So it was by using different chemical or physical factors, was discovered that before activation ionic channels, there is a quantum mechanical sensitive mechanism which generate, which protect activation ionic channels in the membrane. So it's realizing by metabolic deriving water from the cell. And so it was demonstrated that the metabolic deriving water efflux from the cell is fundamental mechanism controlling semi-permeable properties of the membrane, the dysfunction of which is cell pathology. In case in neuron and muscle, if this will have sustainable characters, it will be sustainable excitation, but non-excitable cell also has changing its metabolic activity of the cell. This data and suggestion is close harmony, discovering Lefkovich and Kabilka, who have a Nobel Prize, who discovered that in cell membrane there is a G proteins family, which the activation of which can be realized by even light or other weak signal, and producing intracellular messenger system in the cell, which could, cyclic AMP and cyclic GMP is a very multi-site modulation effect on cell metabolism. To demo in this figure, you can see that extremely low concentration acetylcholine could again modulate gunk-induced current, is voltage clamp experiment, and this modulation is not channel induced, and if holding potential from minus 50 millivolt, we will increase minus 65, and to reverse this response, again, we have inhibitory effect on this. And finally, if we will measure intracellular messenger system, you can see this horizontal dashed line is cyclic IMP concentration in normal state, and 10 minus 4 molar high concentration have no changing cyclic IMP content in neuron, while low concentration increasing it. And finally, when to demonstrate that it's not this modulation channel activity, it's not connect with receptor proteins, by genetic engineering is very easy to demonstrate in oocyte, xenopus oocyte, if we will inject different messenger, specific messenger RNA for different receptors, you can see that they have reversal potential, all characteristic of acetylcholine receptors, but low concentration transmitter have no effect on this. So by this was proved that in membrane there is a G protein, as demonstrated by Nobel laureate Kovatek and Levkovich, that this G protein modulate channel activity. Next, now question is rising, but which is the extremely sensitive mechanism which switch on intracellular cyclic nucleotide, and especially GMP system, because previously we have shown that GMP system more incomparable, more sensitive to environmental factors than cyclic IMP systems. On left side, you can see the cell volume changing by fluidity increasing membrane by using special desanoic acid. It was shown in collaboration our colleagues from Japan that desanoic acid not metabolizing, but 40% increasing membrane fluidity, and membrane fluidity increasing, uptake water increasing, and we have cell swelling and returning back, and you can see that this is discrete changing of cell volume. And right side, you can see that increasing hydration brings to increase GMP content in the cell, and it was shown that soluble guanylyl cyclase is a extra quantitative sensitive mechanism which responds every changing cell hydration. And to demonstrate that brain cortex hydration is a very sensitive to quantum sensitive signal, like as non-thermal intensity microwave. On right side, you can see this again dose dependent by effect on cortex hydration, and you can see that picomole concentration has slight dehydration effect on this. When we use left side, you can see this intensity microwave. It's gigahertz, SAR, specific absorption rate. It's rather weak, which during 10 minutes, no changing water temperature, and treating this physiological solution, and after injection, interperitoneal injection, you can see that this ovine-induced dehydration dramatically increasing. This is just a physiological solution treated by non-thermal microwave. And we have shown that this intensity could change peroxide concentration in medium. So now let's return back again. What happens when intracellular messenger systems are activating? Left side, you can see calcium take in normal state, young and old rats cortex, 10-9 ovain, which activate cyclic IMP content. You can see there is an increasing calcium take, and this is 10-4 also, but 10-4 it's a pump inactivating, and 10-9 before this I have shown that has no direct effect on pump activity. Right side, you can see hydration of cortex in normal, in young animals, and 10-9 and 10-4. So, and pain threshold, you can see that is a normal with PS injection. This is 10-4 injection and 10-9 injection. And this is pain sensitivity 10-9 higher than 10-4. This is pain threshold changing by injection 40 non-radioactive isotope containing medium and 45 radioactive calcium. It's traditionally for study calcium exchange using calcium 45. But you can see that calcium 45 has a different effect than calcium 40. All these data now demonstrate that if we have quantum mechanical sensitive mechanism in membrane, by using this quantum sensitive mechanism can be used therapeutic purposes. And this is a physiological solution, again, 4 Hertz mechanical vibration treated when decreasing peroxide concentration in blood. And you can see there is an increasing pain threshold, pain relief effect. And pain relief effect can be used in case when we have 10 minus 9 molar when cyclic IMP activated. Sodium calcium exchange reverse mode has a pain generation effect. Again, we can remove this, have pain relief effect by 4 hertz mechanical vibration. It's same 4 hertz. All these data clearly indicate that quantum mechanical sensor is O-buying independent mechanism. It's O-buying non-canonic receptors, and by activation, weak signal, chemical and physical signal can be used as a powerful tool for pain therapy, and of course for some disease therapy also for correction dysfunction of cell function. Thank you for your attention. If you have question, please welcome. Every